Epidemiology. This is Chapter 14a, Principles of Disease in Epidemiology. In this chapter, we're going to be talking about pathology and the causative agents of disease. Pathology is defined as the study of disease, and we're going to spend a lot of time looking at the etiology, the cause of disease. So we'll take a look at signs and symptoms and use them to work backwards and talk about what the causative agent is. In that, we'll understand the development of the disease, the pathology, and think about what that means to have that particular infection. What are the outcomes for that type of disease? So, what are the objectives of pathology? It's really about understanding the holistic approach. So thinking about what the signs and symptoms mean, how those relate to the infectious organism, and so studying how disease develops through the changes that the body is experiencing because of that infection. So we're going to talk about normal and transient organisms. We talked a little bit about them previously, but try to understand their involvement both in disease and in health. Transient microbiota are ones that colonize us only for a short duration. This may be for days, weeks, or months, but the real problem with these is that most are potential pathogens. These are the organisms that we pick up throughout our daily lives. Our normal microbiota are the ones that permanently colonize us. They are beneficial to us for the fact that they help to prevent other organisms from colonizing. Typical human harbors about 10 to the 14th bacterial cells. That means that if you look, bacterial cells actually outnumber human cells by a 10-to-1 ratio. We get exposed to our first contact with microorganisms, mostly as we are born. Newborns, those that are born vaginally, their first contact is through the lactobacillus in the vagina to help establish that good normal flora. But regardless of where we get those microbes, we develop a symbiosis. We have to live with them in and on us, and they are important for our health, but we can also see at times they can cause disease. So establishing our normal flora has been an interesting study. So there was a 2016 study out of NYU where they were looking at what happens when you're not born vaginally. So if you were born by C-section, you're not going to pick up the microbes that occur during the birthing process. There's an interesting HHMI video posted, the microbes from mom, vaginal birth versus C-section, that you might want to take a look at. It's been a little controversial. Certainly, we do establish our good normal flora, again, that lactobacillus during our route through the vagina during birth. However, the practice of doing what's been referred to as vaginal seeding has not necessarily been one that has been universally adopted. This is where it has been proposed that we should be doing a microbial wipe to be able to take the vaginal organisms and put those on the baby, both skin, mouth, establishing all the normal flora that they need. As I said, when we see babies are born in a womb or during gestation or in a womb that is sterile. So it's not until they are going through the birthing canal that they start to pick up what is going to be their resident flora. So we see that the flora resembling that of the adult is usually achieved by about 3 years of age. However, there's many postnatal factors, where you breastfed or not, where you live, family members and the organisms that they expose you to. So lots of different interactions are going to determine what your normal microbiota is going to be. So when we think about our normal microbiota, we think about the variety of relationships. Commensalism is a relationship where one organism is benefited and the other is unaffected. Much of our normal flora are commensals. So things like the Corynebacteria in the eye, all the saprophytic microbacteria that are on our ears and our genitalia, just many things that we harbor. There are some organisms that we truly believe are mutualistic. So in mutualism, both organisms are benefited. When we talk about things like E. coli in the gut, they synthesize vitamin K and some vitamin B vitamins for us, and therefore they truly are benefiting us and giving us those vitamins. Parasitism is one where the organism, one is benefited at the expense of the other. For us, we think about this as the disease process, so that when we have an organism that is causing disease, they are being benefited at our expense. Although life is possible without normal residential flora, they are indispensable, really, in maintaining our health and our normal function. And so that's why we have started to question, are these organisms really commensals, or are they more mutualistic? So we can find organisms just in various regions of the body, in the nasal passages, all the upper respiratory, in the stomach, in the small intestine. And without them, we would be susceptible to a lot more infectious agents. When we talk about the normal microbiota and the locations where they are found, we talk about the traditional in things that are found in the upper respiratory, in the nose and the throat. The mouth has just a plethora of organisms. There's many organisms on the skin, the large intestine, the uro and genital system, the lower urethra in both sexes, the vagina in females. However, one of the things that you might notice is that in this traditional picture, as you go down the trachea and esophagus, you do not see flora. You do not see flora in the lungs. You don't traditionally see it in the stomach and the small intestine. And this has been questioned from the standpoint that there is certainly not culturable bacteria there that we can readily get to. However, with a lot of the new molecular techniques, we are realizing that there are much more flora than what we had previously anticipated. Much of this has come out of the Human Microbiome Project by the National Institute of Health that was started in 2008. The goal was the understanding of the microbial flora involved in human health and disease, but we've learned so much about the microbes that are there. These microbes aid in the proper utilization of our nutrients, makes sense for some of our gut microflora, but they also prevent the colonization of pathogens. Beyond that, we have now found that they stimulate the development of certain tissues involved in the immune system, the endocrine system, central nervous system, and you've probably heard a lot recently about the gut-brain axis. When we talk about the intestinal system, that has probably been one of the best characterized for what's going on in both health and disease for our intestinal microbiota. So in health, we have a variety, a large diversity of organisms, and that actually changes a little bit over age. But we see that the balance between what is a healthy and what is a non-healthy gut probably involves what is referred to as dysbiosis. It's not exactly that there's one causative organism. It's usually a lack of diversity and dysfunction of the gut microbiome that causes disease. So we see some of the traditional intestinal diseases that maybe you think about, things like the inflammatory bowel diseases, Crohn's disease, ulcerative colitis. These seem to be changes in populations of bacteria. Specifically, we often talk about the ratio of the firmicutes and the bacteroidetes. If you look down in the lower right-hand corner, you see the configuration for an average Crohn's profile, where we see the collapse of the bacteroidetes and an explosion of the actinobacteria. In ulcerative colitis, we see an explosion of the proteobacteria, a variety of gram-negatives. So even in that inflammatory bowel disease category, we see it's not a one-size-fits-all, but it's the change in diversity of the normal microbiota. We also have realized that this continues into some of the extraintestinal diseases. We have now found links of the intestinal flora makeup with obesity and things like type 2 diabetes. Both of these seem to be associated with a high number of the gut firmicutes. Not only do we see the kind of gut-related diseases there, we've also found autoimmune diseases that we have traditionally known are passed from families, not really in a truly genetic inheritance, but there are genetic factors. But part of this may also be of the inheritance of the family microbiome. So things like lupus, allergies, and asthma seem to have a relationship to the microbiome. And lastly, this true gut-brain axis disorders. We have started to believe that the microbes in the gut may be affecting the autism spectrum, Parkinson's disease, and other mood and chronic pain disorders. So in the news, you may have seen some of the headlines. Unhealthy microbes could promote metabolic disease. That kind of makes sense. Obesity in the human microbiome. Antibiotics weakening progression of Alzheimer's through changes in the gut microbiome. And as I said before, the relation possibly to autism symptoms. In April of last year, there was the reports in a study where autism symptoms were reduced nearly 50% 2 years after having fecal transplant. That is where they're giving the healthy gut microbiome to individuals, and though it's not the cure, it does seem to be helping the symptoms of these diseases. The other thing, do microbes make us sick? Yeah. Do they make us fat? Yeah, that might be true as well. So it is about the diet as well. High fat, high carbohydrate diets favor the outgrowth of these firmicutes, and so therefore we see the change in that gut microbiome. So if you look at the profile of an obese mouse, it is very obese. It would not survive out in nature, but we see that it has an increase in these firmicutes, and that could be contributing to the obesity. This is in comparison to the lean mouse, which has more of the bacteroidetes. So in studies looking at the difference between lean and obese, we see a normal mouse, if they are fed a high-fat or high carbohydrate diet, and they have a normal functional mucosal immune system, we start to see the change in the number of the firmicutes to bacteroidetes. The firmicutes are increasing, the bacteroidetes are dropping, and we see weight gain, which can lead to obesity. If you take a immune deficient mouse and you put them on the same high-fat, high-carbohydrate diet, their mucosal immune response is very limited, and we don't see these gut changes in the microbiome, and we don't see the subsequent weight gain. So we don't completely understand the relationship, but we do know that changes in the ratio of these firmicutes and the bacteroidetes do seem to have a profound role in things like obesity and a number of other of these disorders. That brings us to what was in the autism study, that they saw improvement in symptoms due to what is a fecal microbiota transplantation, or FMT. This has been most successful in treatment of Clostridium difficile, or C. diff. In Clostridium difficile, typically what happens is that people are on antibiotic therapy, it wipes out the good normal flora, and the C. diff is allowed to proliferate. It causes anything from a mild enteritis to something that can be life-threatening, and it's very, very hard to get rid of. Putting them on other antibiotics to get rid of the C. diff is only contributing more to the problem. So they've had good success with doing these fecal transfers using normal fecal flora from a donor. There is not any better way to repopulate the gut microbiome. You can't just give them a cocktail of, oh, here's some E. coli, here's some Enterobacter, the good normal flora. It's something about the balance. They can do this either by enema, a nasogastric tube, or they've also developed it in capsule form, so these poop pills can be taken to help to reestablish the good normal flora, and that resolves the C. diff infection. Through the Human Microbiome Project, we have just been amazed at the number of different species that are present. So when we start to look at places like the intestine, we see that there's anywhere from 500 to over 1,000 different species there that we never even anticipated. On the skin, there can be over 1,000 different species. So when we talk about things like the staph and streps and lactobacillus, this is really just the tip of the iceberg. On the right-hand side in the diagram, you see some of the changes or some of the establishment of the various normal flora in the various areas. So as we said, the vagina is highly populated with lactobacillus. So that's why during the birthing process, that is a major first organism that the baby is being exposed to. When we talk about things, for example, in the mouth, we start to see a large number of the streptococci. So some of the organisms that we've been talking about, those are normal flora in various places, but it's the reality of the Human Microbiome Project that there are so many organisms that we really have not even begun to think about that are part of our normal health and the disease process. So it's also a relationship to our diet. I know a lot of people are always looking for what's that magic microbe that's going to make us have that good, healthy gut, keep us lean, and all those good things. Well, there's no one thing, but one of the things that has been established, people have always been questioning that a vegetarian diet might be more healthy. Well, it's really about the diversity. So even if you're a vegetarian, if you're eating the same thing over and over again, then you're really not making that nice, diverse population. So it could be that diets such as the Mediterranean diet with its kaleidoscope of fruit and vegetables and nuts and grains and legumes really are helping to establish the diversity, which we're finding is more important than necessarily exactly which organisms are there. So if we look at the normal flora chart here from the book, we start to see some of the names that you recognize. So the skin has most of the microbes that we get from direct contact. They're not only on our skin, they're in our pores, in our oil glands. So things like Propionibacterium acnes, the staph, Corynebacterium, those pretty much continue into the eye as well. The nose and throat, you continue to see some of the staphs, but we start to see a variety of what are referred to as the diphtheroids, small gram-positive rods, the streptococci, but even things like Haemophilus and Neisseria. The mouth has a wide diversity. The abundant moisture, warmth, and the constant presence of food makes this hospitable to a variety of streps and lactobacillus, Corynebacteriums, and even fungi. The large intestine, we just talked about things like the bacteroidetes and the firmicutes. Here we see there's a variety of lactobacillus, E. coli and Enterobacter, Citrobacters, the things that we normally find when we played out on MacConkey's and EMB. And in the urogenital system, things like our staphs, our micrococci, enterococci, and a wide variety of organisms. The unfortunate thing, though, is that even though these are good normal flora, some of them can be opportunistic pathogens. If in the right situation, they can cause disease. One of the best examples is the story with pneumocystis. The pneumocystis jirovecii was part of the AIDS epidemic. We didn't really talk about this kind of rare pneumonia until we started to see individuals that were severely immunocompromised because of their HIV status and their progression to AIDS. We also see things like Neisseria meningitidis, Streptococcus pneumoniae, Haemophilus influenza, the Hib. These can be causative agents of things like meningitis and pneumonia that normally they are normal flora. but are kept in check by others. However, changes in the microbial population or other things that may change inflammation, these can now allow these opportunistic pathogens to increase and cause disease. The more classical ones, we just talked about the Clostridium difficile in the intestine, and this is usually what happens with common yeast infection. Whether it is because of the change in pH, because of a woman's menstrual cycle, we often see the overgrowth of the things like Candida albicans causing those yeast infections. They are normal flora, but given the right opportunity, they can be an opportunistic pathogen. So much of our normal flora is keeping us healthy by microbial antagonism. It's competition between microbes. So it is helping to prevent those transient organisms from taking hold. So simply by occupying the niches, those pathogens can't get in to be able to establish and colonize. It's not simply just that I've occupied this territory. Much of our microbiota is also producing acids, which is inhospitable for many of the pathogens, and antimicrobial peptides, things like these bacteriocins that are going to actually kill some of the other microbes around them. We've demonstrated how important this is by using germ-free animals. In an example, germ-free animals can be infected by only 10 salmonella rods, so it only takes 10 of them to cause disease. While the infectious dose for a conventional animal that has its good, normal, healthy microbiota, you need about 1 million cells before you actually get the disease from the salmonella. That is why we've seen a lot of interest in using probiotics. Live microbes, whether they're applied or ingested, to try to promote those beneficial effects. So things like lactobacillus is always a good one, but we also talk about various streptococci, the Thermophilus, and E. coli. In the right proportions, these can help establish good, if it's got normal flora, to be able to prevent disease. Interestingly, we also help out in this. We have barriers to infection as well. Our epithelial cells are actually producing antimicrobial peptides. In an interesting study, they found that one category of what's referred to as these S100 proteins, that's because they are only about 100 amino acids long, the psoriacins are able to selectively inhibit certain microbes. In a study where they painted on Staph aureus on people's fingertips and E. coli on other fingertips, and then allowed that to incubate on their fingertips for 30 minutes. They then asked them to press their fingertips to agar to see what grew. The psoriacins actually killed off the E. coli, but Staph aureus was able to grow. So it's interesting that these antimicrobial peptides are allowing some of the normal flora to still flourish, but we don't really want E. coli on our skin. It should be in the intestine. It's a fecal contaminant. And so our antimicrobial peptides from our skin layers are actually helping in that regard, so the E. coli can't get established on the skin. So that's why it's important for us to understand our human biome. We are really 99% microbes, and so understanding what they mean for our health and in disease is important. We'll spend a good portion of the last part of the quarter talking about the etiology of infectious diseases. Robert Koch was the first one who established Koch's postulates in saying this is the causative agent, so kind of a one-for-one of this organism is causing this disease. If we look at Koch's postulates, they have been used to prove the cause of infectious diseases, first published back in 1890. So you first have to start with a diseased animal. It doesn't have to be a dead animal, but it has to have some sort of microorganism that caused the disease. In the lab, we would have been doing a lot of growing of various organisms in pure culture. So in the disease process, we have to be able to get this organism growing in pure culture and then identify it underneath the microscope, do the biochemical testing to establish the identity of the organism. The microorganism then needs to be reintroduced into a healthy lab animal, and the disease needs to be reproduced in that lab animal. If it was a respiratory disease the first time, it has to be a respiratory disease now. And we then take that disease animal that has received that pure culture of that organism, and we now have to re-isolate that organism. So the same microorganism that we isolated from that first animal, we have to be able to isolate it from the second animal and grow it again in pure culture and establish its identity, both by underneath the microscope, gram staining, and our various biochemical testing to actually establish that that is the cause of that disease. So that's why Koch's postulates are so important. They rely on pure culture to be able to establish what is the causative agent. We have actually also updated Koch's postulates to include what is referred to as the molecular Koch postulates. So we know that there are some things that we're identifying by more molecular techniques rather than doing things exclusively in culture. As with everything, there's got to be exceptions to the rule. So we think of with Koch's postulates that we have a disease and there's one organism that is the causative agent. And that's typically how we do a differential diagnosis, things that we will do later in the term. However, some pathogens can cause several disease conditions. So if we say somebody has an infection with Streptococcus pyogenes, we don't know if that person has strep throat, scarlet fever, toxic shock syndrome. So that is just too many diseases from that one organism. Some pathogens cause disease only in humans. Neisseria gonorrhoeae, the causative agent of gonorrhea, HIV, these do not have animal models. So if there are no animal models, we have ethical issues in trying to establish that causative agent, even though we accept these as the causative agent for these diseases. We also have some organisms that are just hard to grow in pure culture. Treponema pallidum is the causative agent of syphilis. However, in tissue and fluids, we can use fluorescent antibodies and know that it is the causative agent. Mycobacterium leprae, the causative agent of leprosy. We can't grow it in pure culture, but we can grow it in armadillos. So we can be a little bit creative. Legionnaire's disease that was first found in Philadelphia was very difficult to identify the causative agent because we didn't have the techniques to be able to grow it. We also have issues with multiple organisms causing the same disease. When we say things like pneumonia and meningitis, we start to see that there's not one causative agent for pneumonia, for meningitis. We have to identify the specific causative agent, so that goes a little bit against that one-to-one rule established by Koch's postulates. We're now going to talk about classifying infectious diseases. What does it mean for us to be able to spread disease, ones that aren't communicable? So if we look at classifying infectious diseases, first we need to talk about things like signs and symptoms that go into our identification of our differential diagnosis. A symptom is a change in body function that is felt by a patient as a result of the disease. It's subjective. These are things like pain and fatigue. People can describe them, but one person's pain, if they rate it at a level 8, might be somebody else's 5. So it's very subjective to that individual. A sign, on the other hand, is a change in the body that can be measured. It's observed as a result of disease. So it's something that a clinician can measure. These are objective changes. We can measure vital signs. We can measure fever, paralysis, swelling. And therefore, these are going to be things that are observed and, again, are going to be objective by the standard of what is measured. When we put those all together, we refer to that as a syndrome. Not everything is passed from one person to another. So it is important as potential disease-causing agents are in the environment, what actually gets communicable. So what is spread from one host to another is very important if you are in a clinical setting. So things like colds and flu and whooping cough, these are communicable diseases. Communicable does not have to imply something like a sexually transmitted disease. It doesn't have to be close, intimate contact. It just means that a disease can be spread from one host to another. A special category is contagious disease. We refer to this as those diseases that are easily spread from one host to another. These are things like measles and chickenpox. If you've been exposed and you haven't been vaccinated or you haven't had those diseases, you're probably going to get them because they are so easily spread. It is unfortunate people always kind of interchange those two things, communicable and contagious, but they are slightly different. Non-communicable diseases are things that cannot be transmitted from one host to another. You can't get tetanus from a patient. So they get tetanus because they got endospores into a deep puncture wound. The endospores germinated, the toxin was formed, they start to feel the signs and symptoms of tetanus. Botulism, you get that because you ingest the toxin in food unknowingly. So there's no way that those can be transferred or transmitted to another person. When we talk about the communicable diseases, we rate them based on an R0, and this is the basic reproduction number. So we looked at this before with the viruses, but this is how we're saying measles is contagious. That is because it has a very high R0 number of 16. So it is going to be one that if you come in contact with that person and you are susceptible, you're going to get that disease. So things like measles and chickenpox are notable for the fact that they are highly contagious. We also see occurrence of disease, and we've been talking a lot about herd immunity. So we understand when you have a new and novel pathogen, it's easily spread, as we're currently seeing with the COVID-19 pandemic. We as a population have no immunity, and so therefore it's going to be spread very quickly. Hopefully over time, we're going to start to get some immunity. So as people develop immunity to the disease because they have had the disease, although with the coronavirus, we're not quite sure how long this immunity is going to last, but we're also hoping that we're going to get a vaccine. We don't have one yet, but definitely lots of people working on it. In the end, what we're hoping is to provide herd immunity. That if we have enough people that are immune, either by having had the disease or we're able to get people vaccinated, then the pathogen just doesn't have any new hosts to be able to infect. So the herd immunity takes hold when the pathogen can't find new hosts, and then we stop the spread. So that's what we are hoping. We're not sure what percent of the population is going to have to be established to be immune before this is going to be effective for COVID-19. But we know that we probably are going to need some combination of natural immunity from the disease and vaccination in order to get us out of the COVID-19 problem that we are currently facing. We also talk about occurrence of disease by incidence and prevalence. The incidence is the fraction of a population that contracts a disease during a specific time. It's the indicator of the spread of disease because it's only the new cases. Prevalence, on the other hand, is the fraction of a population having a specific disease at a given time. It is new and old cases, and so is the indicator of how serious a disease is and how long it affects the population. So the little cheat sheet to the bottom left-hand side, prevalence is all cases and incidence is new cases. But we can think about this as a model in our bathtub here, where incidence is what's coming out of the faucet and going into the tub, whereas what's into the tub is prevalence. That is all the old and new cases that are accumulating. You can have some recirculation of recurrence, But we unfortunately think about that the way to get out of there is oftentimes by mortality as people succumb to the disease. When we look at the reported AIDS cases in the United States, we actually saw waves of the classification and the expansion of the AIDS epidemic. So we could talk about this in looking at these different waves by the number of new cases. It's a little hard from the bar graph maybe to get a feel for that, but if you look at the line graph down in the right-hand corner, tracking both prevalence and incidence in the United States of HIV from 1980 to 2010, we see that in the blue line, that's the incidence. That's the new HIV infections. So we had our peaks, our humps in the 80s, and we then started to see that it leveled off. However, if you look at the red line, which is the prevalence, which is the total number of active HIV/AIDS infections, that is what is continuing to climb. We are not curing people of the infection, and that's why we see the AIDS epidemic raging on. So that brings us to other measures of occurrence of disease. We definitely know, because of the COVID-19, what is a pandemic. So we are currently experiencing that worldwide epidemic because of the coronavirus. However, other classifications, we see sporadic disease is disease that occurs occasionally in a population. So maybe you have cholera experiencing somewhere. Endemic diseases, on the other hand, they are constantly present in a population. So they are there. Colds and things are always kind of around, but every once in a while you have different numbers. Epidemic, disease acquired by many hosts in a given area in a short time. So we can see both in looking at the maps of North and South America what that might mean for the difference between endemic, sporadic, and epidemic. Epidemic, you see that cluster. Pandemic, it's spread over multiple continents. But we can also look graphically. When you look at sporadic, it's the lower blue line. It's just kind of always there, and sometimes you have a little bit of an increase. Whereas the top line in green is the endemic. This is going to be something where it's a occurrence that is there in the population at all times, compared to the peak of an epidemic where you have a spike in numbers. We also can look at the severity or duration of disease. We are much better equipped in medicine to think about acute disease. Symptoms develop rapidly, so we're thinking of here's the disease, it presents signs and symptoms, and then we get rid of it, whether that be by intervention or our immune system. Chronic diseases are problematic. We get the disease and then it just kind of becomes a slow lingering. It may decrease in its severity, but it just hangs on and becomes chronic. Subacute, something between acute and chronic, is a little bit harder to define, but it usually is somewhat under our radar. And latent diseases, as we talked about before, herpes, a classic example. You have peaks and valleys of latent infections. So periods where there's no symptoms when the patient is inactive, but the disease can come out of remission and have active signs and symptoms as well. Other definitions? Most of the time when you get an infection, it's local. So it's in the respiratory system or a cut that gets infected. So local infections, pathogens are contained to a limited or small area of the body. Systemic infections are obviously more problematic. This is infection throughout the body. This often occurs via focal infections. Focal infections are when you get systemic infections that begin locally, but oftentimes travel via the blood to cause a systemic infection. Blood is meant to be sterile. We can have a little bit of transient bacteremia, bacteria in the blood, but white blood cells clear it out very quickly. When somebody goes into septicemia, that means that you actively have growth of the bacteria in the blood. Remember, iron is something very important for the bacteria to get to be able to grow. We also can measure toxins in the blood, toxemia, viruses in the blood, viremia. All these things are important depending on the disease-causing agent. Again, extent of the host involvement. Primary infection is what we are used to dealing with. People get an acute infection that causes the initial illness. We experience those signs and symptoms. But oftentimes, people may get a secondary infection. This is an opportunistic infection after the primary infection causes a predisposition, leaving them more susceptible. One classic example, unfortunately, is in our elderly, that they often get pneumonia after a bout of the flu. The flu has compromised their lungs, and now they're more susceptible to getting that bout with pneumonia, and that can often be lethal for them in their elder years. Subclinical diseases are hard to explain. There's no noticeable signs or symptoms, so they're in apparent infection. So they're definitely under our radar. Hepatitis A among children is example, and the other one that you may have seen ads for is hepatitis C in the baby boomers. People that were born between the years of 1946 to 1964, they have a high risk of having hepatitis C and may not even know it. We're going to take a look at the patterns of disease. So how does the disease progress? All diseases are going to have these defined stages. There's always an incubation period where you have been exposed, but you're not experiencing any signs or symptoms. These are going to vary widely. You can have something where maybe it just takes a couple of days before you start to see signs and symptoms. Something like rabies might literally take months before you actually see those signs and symptoms. After the incubation period is the prodromal period. Mild signs and symptoms where you're feeling I feel achy, I feel tired, I kind of have watery eyes and a headache, but nothing descript. The period of illness, most severe signs and symptoms, and that's usually what defines the disease. Hopefully at some point, whether it's intervention or your immune system kicking in, we start to level off and then go into the period of decline. Signs and symptoms begin to decrease, and then hopefully we enter the period of convalescence where, again, we recover. We can potentially be infectious in any one of these stages. It is disease specific. So there are some diseases where people may actually be contagious in an incubation period or a prodromal period and not know it. We can see people that could be infectious during the period of convalescence. So it is definitely disease specific. When we talk about disease, there are many things that contribute to disease. Predisposing factors can be so many things. They just make your body more susceptible to the disease. Sometimes it's anatomical. Things like a short urethra in females makes them more susceptible to urinary tract infections. Inherited traits with sickle cell. These individuals are more susceptible to pneumonia, blood infections, meningitis, and bone infections. We also see changes in climate and weather can cause us to be predisposed to various diseases. And more individually, our age, our status, whether maybe we're undergoing chemotherapy, or our lifestyle. Overly tired means that we're often susceptible to disease, and stress. The relationship of stress to our endocrine system impacts our immune system. So it is not all in our head that when we're stressed, we tend to get sick. And that's why it's important, even though when we're feeling tired and stressed, that we do need to take time to re-energize because we don't want to get sick because of these predisposing factors. Make sure you check your understanding, and we'll look at more of our disease and epidemiology in Part B.